Hello and welcome to Nikolai's genetics lessons, and today's question is: the template strand of the gene being transcribed is as follows. What will be the sequence of the RNA made from this template? If you think that this is a very easy question, please watch the video, because I'm going to explain today something you probably didn't know before, or which caused a lot of confusion for many students. So here is transcription bubble. This is double-stranded DNA, and I will explain why I'm showing these two strands of the DNA in different color, and why I'm showing this messenger RNA that is being made in the same color as the top strand of this double-stranded DNA. Do you know, by the way, what prime ends are going to be here on this double-stranded DNA? You may say it can be any, but this is not so. Take a look. Messenger RNA always is made from 5' end to 3' end, only in this direction. So that's why I know that this is 5' end. And I also know that this is template strand of the DNA. This messenger RNA is going to be complementary to this strand. That means that this strand of the double-stranded DNA have to be 3' end, and this one 5' end. And also we would have 5' end here and 3' end here. You see that 5' end of the messenger RNA and 5' end of this strand of the DNA is the same, in the same direction. This is not the only reason I'm showing messenger RNA and this strand in the same color. I will give you explanation a little bit later. Now let's put these nucleotides that's shown here on the template strand of the DNA. So the sequence is going to be C, T, T, G, C, C, A, G, and T. So the sequence on the corresponding messenger RNA that is being made using this template is going to be, so all you need to know that thymine base pair with adenine and guanine base pair with cytosine. But in messenger RNA, in RNA, thymine would be substituted with uracil. The sequence is going to be as follows. So guanine, adenine, adenine, cytosine, guanine, guanine, uracil, cytosine, and adenine. And what is going to be sequence on this strand of the DNA? And the sequence is going to be the same as in messenger RNA, with only one difference, that instead of uracil we would have thymine. So the sequence is going to be guanine, adenine, adenine, cytosine, guanine, guanine, thymine, cytosine, and adenine. Because sequence of the messenger RNA is the same as this strand of the DNA, we call this strand of the DNA coding strand of the DNA, coding. And that's why we call this strand of the DNA non-coding. You also know that we call this strand also template strand of the DNA. That's why we call this strand of the DNA non-template strand of the DNA. We also say that this strand of the DNA have sense, so sense strand of the DNA, and this is going to be nonsense strand of the DNA. We also call this strand positive strand, and we call this strand negative strand of the DNA. So I hope now my explanation throw a light how strands of the DNA are called. And sometimes you also can find that some scientists would say that this is top strand of the DNA and this is bottom strand of the DNA, but this is not scientific name, this is just in reference to certain picture. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day, and see you in the next video. Goodbye.